Welcome to the Maine Science Podcast. I'm Kate Dickerson. Today's episode is a conversation I had with Rob Wheeler, Associate Professor of Microbiology at the University of Maine. Rob studies fungus from both sides, how the immune system responds to fungi infection, and how the fungus itself works. Our conversation was fascinating, and it was great to learn more about an area of science that I know little about. I hope you enjoy our conversation as much as I did. Well, Rob, welcome to the Maine Science Podcast. It is a delight to talk to you. I know a little bit about your background, but not a ton. So before we dive into the amazing world of fungus, I was hoping you could talk a little bit about where you grew up and how you got into science.
Yeah, so I'm one of those people that hit their scientist age at age 2 and never really stopped asking why. I went through periods where I was not quite sure what the questions were that I wanted to ask, but I still have so many more questions every day than I have answers or ways to answer them that I really get entertained on a daily, weekly basis. And it's always hard to decide what to focus on for research that we do in the lab. I grew up outside of Pennsylvania in sort of Quaker country, not really in the countryside, but more in a suburb. And I have vowed to avoid suburbs at all costs since then. I've lived either in, smallish cities like Cambridge, Massachusetts, or larger cities like Munich, Germany, and now a pretty small town or a large village of Orono, Maine. So I was always excited about the outdoors, and really that was what got me interested in science initially. Of course, growing up in the 80s, there was this tremendous push towards molecular biology that was happening, and I really got caught up in that as well and got excited about what we could learn about the natural world by understanding the genes and molecules that really play big roles in that. So when I was in college, I really fought with myself about what career choice I might do. It was both science, but I really fought between field biology and molecular biology, and I ended up with molecular biology, but I still have a lot of interest and excitement about field biology.
So just for people who don't know the difference, would you mind doing just a super quick overview of the difference between field and molecular?
Yeah, so just to give you a sense for one specific event, so what my sort of peak in my interest in field biology was when I volunteered for the U.S. Fish and Wildlife Service, and I worked on the Big Island of Hawaii to study bird populations of endangered bird species. So we were walking transects in the forest and looking for bird nests part of the time. Part of the time we were mist netting for birds and extracting them from the mist net and banding them and weighing them and measuring them and so on, which was absolutely an amazing experience. And then we also did a little bit of tracking of birds with radio transmitters, where we would set up by ourselves in a tent on the top of a hill for a week and just track birds every hour during the daylight hours with a little radar dish. So that's sort of one example of a field biology experience. And then the molecular biology side, I studied a number of different things, but we could just, as one example, we could look at something that I studied as a PhD student, which is how different proteins that regulate both cell cycle, how the cell decides to proliferate. And those proteins also decide whether a cell turns into a mobile or a stationary adhesive cell. And we studied how those proteins move around in the cells during the cell division cycle as the cells grow and then divide.
So was the bird research when you were an undergrad? Was that part of the
yeah.
Kind of figuring out what's a good path for you type of thing?
Absolutely, yeah. I was really fortunate to find this group called the Student Conservation Association, which is still around and provides a tremendous database of jobs for people who are interested in conservation biology and the environment. And I said, well, if there's one summer job that I'd love to do, I'd love to be doing field biology on Hawaii, and somehow it worked out.
That's pretty great. I certainly realized when I was in college the value of summer jobs, of winnowing out what is good and what isn't for you. And I think it's something that we don't talk about enough, that it's really good sometimes to have a job that you know is not how you want to go. And you just tough it out for those, couple of months and then move on.
Yeah, it's funny. In this case, I loved what I was doing, but the valuable experience for me was to meet those full-time career biologists for the Fish and Wildlife Service and see what their lives are like as a 40-year-old or a 50-year-old. And I kind of decided that this was a wonderful life for me as a 20-something, but I might not still want to be doing this as a 40 or 50-year-old. And that's just my experience. And I'd love to be able to provide that kind of forward-looking experience for the undergraduates that work in our lab, for instance, that they get to meet the graduate students, the PhD students, and see whether a PhD is something that they would like to do. So they get to see, not just meet them for 10 minutes or 15 minutes, but just really get to see them on a day-to-day basis and see what they do and their frustrations and their joys and so on.
That's a really good point, and an excellent segue into you talking about your, I was going to say current research, but from what I've seen, this has been a long time focus for you, fungal research, and I am not going to embarrass myself by trying to explain it. So I'm going to sit back and hand over the microphone, so to speak, and periodically interject if I have questions.
Yeah, so we study this one fungus called Candida albicans. Most people are familiar with Candida because it's the main cause of yeast infections in women, and that's the one disease that it really causes in healthy individuals. But unfortunately, there are some people that will know it because they know a loved one who was in the hospital and got a Candida albicans infection, which is the second kind of most common place to get a Candida infection. But unfortunately, in the hospital, those infections tend to be bloodstream system-wide spread infections. And unfortunately, we don't have very many drugs to treat those invasive fungal infections that people get in hospitals. So it's one of the main reasons why people die in hospitals, unfortunately. So people have heard of MRSA and other antimicrobial resistant bacteria that people will get in the hospital. And those antifungal resistant fungi and difficult to treat fungi are also a major cause of disease. So unfortunately, in bone marrow transplant patients, that's one of the biggest causes of mortality. So it's a real driver for understanding how the fungus causes disease, how our immune system normally deals with that infection, and how we might be able to exploit that information to find better ways to diagnose disease, who has the disease, and figure out new methods, new strategies for either helping the immune system out or killing the fungus. So we study both sides of that kind of equation, both the fungus and the immune system, because we've realized that if the immune system is down, then it provides more opportunities for the fungus to cause dangerous disease. And if the fungus has one virulence factor or another, then it can resist the immune system.
So I am actually going to interrupt quickly. Sorry. How is it this causes relatively common and problematic, but not life-threatening yeast infections with women? And then when it becomes, I'm sure you get this question a lot, I can tell by you nodding, like, it's interesting to me that it is so deadly in the bloodstream, but how does it even happen?
Yeah, that's a fantastic question. And it's something that is really a mystery. I think I would say it's still a mystery today. What we think of, so we've actually a very new avenue of research. I mean, it may, for the layperson, it seems like, somebody who studies Candida albicans for 20 plus years is focused on this really esoteric subject. And it's kind of one thing, but really there's lots of different things within that field that we've studied. And one of my current passions is actually understanding the vulvovaginal candidiasis, that is the yeast infections in women. That's kind of the more scientific name for it. And probably because people have issues saying words associated with yeast infections and human anatomy, and also because women have been a marginalized population in terms of understanding health and disease, this particular disease has really not received as much attention as it should, because it really affects women worldwide and it can cause significant short-term consequences. And some women get chronic disease as well. Actually, one of the really interesting things is that it's the most accessible form of candidiasis to study, because we can look at it in healthy patients, and to look at what's happening in the disease, we don't have to do very much invasive work. And when we have, a person who has Candida in their brain, we're obviously not going to take pieces of their brain out and start looking at it. But with VVC, vulvovaginal candidiasis, we can really study it directly. And so by studying it in collaboration with a really wonderful group over in Italy that we've been working with, we've been really trying to ask this question, which is, why is it that some women get this disease and other women don't? And for a long time, this has really seemed to be blame the woman. Okay. So there's something about women who get the disease which predisposes them for disease. And the women who don't get it, there's something about them that is protective about them. And, I say that in a little bit of, humor, but there is some truth to it. So we know that there's some truth to it. We know that there are some immune genes that specific variants of those immune genes are associated with women who get chronic disease, chronic yeast infections. And those changes to our immune system actually are changes that turn up our immune system. So it seems like women who have an immune system that's very good at fighting disease also have a greater propensity to have stronger inflammatory reactions to organisms that might normally just be tolerated by our immune system. And those are women that tend to get more symptomatic infection. So because we know a little bit that it can be associated with human immune changes in our genes, we as a group, I would say, as a community of infectious disease scientists, have focused a lot on the human side of things. But this collaborator and I really started looking at the fungus side of things to ask the question, are there some strains of Candida that are able to cause disease and other strains that aren't able to cause disease? And can we figure out what's different between those groups of Candida strains? There are lots of different Candida strains out there in the world. Is there something about them that's different? So we actually found that there is a big difference between strains that come from women who don't have any disease, but just have what we call Candida carriage. They have it, but don't have any symptoms, and women who actually have disease where they have itching and burning and so on. And it turns out that there's a disease symptom, which is this sort of white stuff that comes out, which is made up of human cells and Candida cells. And we can model that in a petri dish. And the commensal carriage strains kind of don't cause as much of that as the symptomatic disease-causing strains. So this is the first time in decades of work to understand what's happening between the fungus and the host in vulvovaginal candidiasis to really give us a sense that maybe there's something in the fungus itself in those particular strains makes them able to cause disease. So it seems like part of the answer to your question is there are differences from one human being to another. And that can be in the genes. It can also be in environmental factors. But there are also things that happen on the pathogen side where some particular strains of a pathogen can cause more disease than others.
Do you have any real sense of how many different strains of Candida there are?
No, I mean, we know that there are hundreds because we've sequenced them as a community. But I'm sure there are tens of thousands of strains of Candida out there in the wild.
I'm thinking that that's maybe one of the reasons no one thought about this, because it's got to be so hard, right? Because have you found new strains while you've been doing this? Let's look at the fungus thing.
Well, absolutely. And, we can define, sometimes they're very closely interrelated strains and so on. But yeah, this is a huge new area that has come out really from technological advances and being able to get the complete DNA sequence for individual fungal strains and process that data and then look at the differences and similarities from one strain to another. And this is really a huge sort of Wild West, I would say, of our understanding of the disease. Now, every year, hundreds and hundreds of strains are being sequenced and analyzed. And this is telling us more and more about how this fungus can cause disease.
So are you finding this overwhelming? And when did you realize in the span doing this work that holy cow, there's tens of thousands of these?
Yeah, I mean, it's kind of, in one sense, it's overwhelming, but in another sense, it's super exciting because there's all this variation out there, this natural variation out there, and we can use that natural variation in order to understand human disease, just the same as we can understand human disease from the human perspective at looking at the natural variation in human beings. So on one sense, it's overwhelming, but on the other sense, it's super exciting. And we know that there are some things about the fungus that are conserved among all of the different strains. So we can use those as sort of a baseline, and then we can look at individual characteristics of them to identify what might be causing disease.
So is it analogous to how the COVID virus evolved so that there's, like a core part of it that was the same, and then there's just these we still have the spike protein, but there's these other things. It's the same idea as that?
That is absolutely the same idea. In any individual infection, the viruses make lots and lots of changes, even in one individual over the course of an infection. And then every once in a while, those changes make the virus more virulent in some way. Either it evades the immune system or causes more disease or in some way or infects a different kind of tissue, something like that. And we can think of those little tiny changes that happen in an individual as sort of closely related Candida strains. And then we can think of those big jumps as maybe the difference between one group of Candida strains and another group of Candida strains. And one may be able to cause disease in a more virulent way than another group. But
okay, so now,
because with the viruses, we know we have sequences from hundreds of thousands of viruses. So we're still in the early days for the Candida.
I have two questions. This first one is why Candida? What was it about this fungus in particular? And then secondly, could you step us through at a basic level what it means to be doing research in your lab on this fungus?
Sure. Yeah. So let's see, two pretty different questions. So why Candida? So that's actually, there's some scientific things about that and some personal things about that kind of. So when I was finishing my PhD, I was looking for something exciting to do with my career. I knew that one thing that I was excited about was infectious disease, but I also knew that I wanted to study something that was not bacteria. So I've been studying bacteria as a PhD student, and I wanted to study a different type of infectious disease. But at the same time, I had fallen in love with molecular genetics. I loved the idea that you could find out what individual genes do by changing them around in the pathogen. And when I was looking, there was really only one non-bacterial pathogen that one researcher could work with, could actually find out the function of individual genes. That's changed now, but at the time that was it. Also, there was a really fantastic mentor that I met, this guy named Jerry Fink, who is still a little bit at the Whitehead Institute. And he was a trailblazer in the molecular genetics of yeasts. And since Candida is kind of a yeast, he had really moved into that type of infectious disease. So that's kind of why I started getting into it. But another part of why I started actually was because when I went to visit the lab, the people that were there working in the lab, I just really got along with. I thought that they were really exciting people to spend time with. They had different perspectives on life and were really nice and fun people. And then also scientifically, they were really the kind of people that I liked. They were really interested in the science. They really wanted to push the science forward. They didn't care about fame or fortune. They were just excited about pushing the science forward. And why did I stay? I stayed in this really esoteric field of Candida albicans biology because the larger worldwide community is also fantastic. I love to go to meetings with people that study Candida albicans and other fungal pathogens. There are a number of other fungal pathogens that are really important as well worldwide. They are the best group of collaborators and scientists. I love it. We share reagents, we share ideas, we collaborate with each other on projects. We have, in my lab, developed techniques in order to look by fluorescence microscopy at what's happening between an individual Candida cell and an individual immune cell in a transparent zebrafish, which is a vertebrate host. And other people that are working on mice or working in petri dishes love the idea that they could actually watch what's happening during an infection of a live host. So that has really provided us with a basis for establishing collaborations with people in Australia and Germany and UK and Italy, et cetera. So it's been a really, really fantastic global community that I've been involved in really since 2003 or so, 2004. And that's really what sustains me. And your second question was, what is a week in the life of a student in the lab? Well, we have all sorts of different projects that are going on. But a lot of the work is, again, involved in watching what happens in a transparent zebrafish between an individual immune cell and an individual Candida albicans cell. So as you might imagine, that's a pretty important thing to understand if we want to understand how infections happen, how the immune system normally keeps this fungus in check. And you really can't do that in a mouse or a human because it's very, very difficult to watch those things happening in such a large organism. And you really can't do that in a petri dish because a petri dish is such a simple system. So in the zebrafish, we can engineer the fish to have fluorescent immune cells like green or red. We can engineer the Candida cells to be making fluorescent proteins in blue or green or red or far red even. And then we can put them together and we can watch what happens over 4, 6, 8, 12, 48 hours between the fungal pathogen and the immune system. So a student who's doing an experiment along those lines, they will spawn some fish that have those fluorescent immune cells. They'll grow those eggs up. They'll check to see that they have the fluorescent immune cells, and then they'll infect the fish with a fluorescent form of the Candida albicans of one type or another, depending on the genes that we're looking at to see what's going on the pathogen side, or they might change the host by using a zebrafish that has a mutation in it. And then they'll look on the confocal microscope to see how many immune cells are there and fighting the Candida infection. Is the Candida growing inside of the immune cells? Is it growing outside of the immune cells? And is it causing damage to the host? And then they'll take those images and they'll quantify those images using different image analysis packages and then come up with real scientific results to understand whether this particular gene in the immune system or this particular gene in the fungus is important for immunity or for pathogenesis.
That was such a great example to me of the iterative process of science and very small steps that get combined to make a large picture. I'm convinced that's one of the reasons I don't have a lot of patience, right? So the lab, my lab life was not great. Like the steps it takes to do that, I think, are extraordinary. And I'm always impressed that people can do that because let's go, which you can't do with science. But it's a really good reminder that you need those little steps to do it.
Absolutely. Yeah. And it's very, very challenging. The real benefit and real treasure that comes out of it can be these beautiful time-lapse movies that you can really watch those immune reaction in what seems like real time. And it provides such an amazing perspective into what's happening in our bodies during an infection and the tremendous power of the immune system to deal with infections on a day-to-day basis.
Rob, I am seeing a whole exhibit of a sort where we can take these films of yours and really explain something. I am going to circle back to that and not this conversation. So I think one thing I want you to explain is why the zebrafish. If I remember correctly, we share 97% of the same genes, which is completely and utterly not intuitive. I actually agree. I've had the good fortune of seeing zebrafish, and they are really amazing with the translucence, but the genetic part is really interesting.
Yeah, so I'll tell you that I'm a sucker for this kind of thing. And the first time I realized that we could use zebrafish as a model for infection, I was just taken in hook, line, and sinker. I had no idea what amazing scientific questions we were going to study. I just wanted to watch it happen on the confocal microscope. I just thought that was the most exciting thing. And then we actually did it, where my student Kim Brothers figured out how to do it. And then we were like, okay, so now what can we do? What can we do with this? So let's take a step back. So why were we excited to use zebrafish as a model? Well, zebrafish are vertebrates. So it means that they have a backbone like we do. And because they have a backbone, they are very closely evolutionarily related to us. They have an immune system that's actually not too different from our own. They have immune cells that are similar, genes and proteins and signals and tissues that are really not that different from our own. And so that provides us with a little bit simpler system where we can ask questions about the types of immune cells that are involved in an interaction in immunity to a particular pathogen. And then we can ask questions about genes as well that are similar. And then we can take those ideas, we can collaborate with others or do experiments ourselves with mouse models of infection to test if those same mechanisms that we see happening in this zebrafish are also relevant for mice or potentially for humans down the line. So there's a tremendous amount of work that's been done using zebrafish as a model for human disease. In addition to 97% of the genes being conserved, it's about 97% of the human disease genes that are also conserved in zebrafish. So there are many, many models of human diseases that have been translated to zebrafish. And we do that a bit as well. So we look at primary immunodeficiencies that are only present in a few human individuals, as they're quite rare, but we can then model those in zebrafish. Yeah. So that's one part of it, which is that the genetics are very similar. But the other part of it is this fascination that I have with being able to watch what happens during an infection. The first week or so of life, zebrafish are really transparent, and they're really small. So we can put a whole fish on a confocal microscope and watch what's happening everywhere in the fish. We can target a specific gene and then see when that gene is turned on and off, which cells it's turned on and off. And that tells us something tremendous about what's happening in the whole organism during an infection. An infection in one area affects something elsewhere in the host. So it provides us with a really global view of that infection.
So at the risk of totally exposing my ignorance, I'm going to assume with the microscope that the fish has to be humanely killed so that you can actually know. I'm totally wrong.
Absolutely not. That's a wonderful question. And that's the most amazing thing is that these fish, they breathe through their skin, basically, when they take up oxygen through their skin. And we can kind of immobilize them with a very soft jelly-like substance called agarose, a really low melt agarose. And we can kind of paralyze them so that they don't twitch. And we can watch them while the fish is still alive for hours and hours. And then we can take the jelly away from them, and then they can swim away and totally survive. So it's a tremendous ability to actually watch what's happening in that living organism over time.
Okay, this is even cooler than I thought. And I can totally see why you wanted to do this. So what has been, what you're doing sounds to me what I have referred to in other conversations as basic research. That's really critical, right? Like, I think one of the problems that people like me have is like, okay, yeah, but how can you apply it? It's an important question, but it's not the only important question. So what you're doing is really laying groundwork. Have you been able to kind of have some, I don't want to say aha moments because that feeds the stereotype of science has to have aha moments, but have you seen the building up of the foundation where you actually can kind of see it, 10 years into the future that there's going to be real concrete results of this basic research that may have only been dreamed about 20 years ago?
Yeah, maybe I'll give you two examples of that. One example was really an aha moment when I was a postdoctoral fellow, and I was really interested in the ability of the Candida to evade immune recognition. There's an immune receptor that is able to bind part of the outer coating of the Candida. And we found that the Candida covers that immune reactive molecule with another coating of a sugar. And that actually prevents the immune system from really recognizing the Candida. And it's an immune evasion technique. And I thought that was really cool. It's really kind of basic science, but I thought it was really cool. And then I had a wonderful conversation with a more senior scientist who had been running her own lab for a while in a totally different field. And she said, well, what's the importance of this? Why aren't you doing this? Why do we care about it? And right on the spot, it just forced me to think about that. And I thought, wow, well, we know that one of the drugs that as used in the clinic to treat fungal infections, including Candida infections, actually changes the ability of the fungus to make this outer layer of it, and therefore would probably affect the ability of the fungus to evade the immune system by making that outer layer. And right there, I thought, oh gosh, I have a set of experiments I can do. And it was the one time in my life, I think, where I went into my supervisor, I said, this is the experiments we're going to do, and this is what they're going to tell us. And I went to the bench, and a few weeks later, I had the drug in my hand, and I did the experiment, and it turned out exactly how I expected it to. When you use the drug at levels that change the fungus feels, but don't kill the fungus at these what called sub-inhibitory doses, less than the level that's required to actually inhibit the growth of the fungus, it totally changes the ability of the fungus to evade the immune system. And that's really important because it means, what it meant is that every time that drug was being used in a human being, it was changing the immune response to the fungal infection. And this is something that the company that marketed the drug and still markets the drug had no idea was happening. Okay, so all of a sudden they got excited about it and they thought, well, if we can identify other ways that drugs can both block the growth of the fungus and also change the immune recognition, then this may be a way of developing better drugs in the future. So that's one of the things that they've been doing since then. So that's one example of really an aha moment, I would say, which comes always, almost always for me in conversations with other scientists where they really push me to the next level. And that's why I love, meetings and so on. And then another thing that I'll give you another example, which is a current example, which is, as I was mentioning, we identified how some strains of Candida are associated with disease and other strains are not associated with disease. So if we can understand that, what that tells us is if we can understand markers that distinguish those two populations, then we may be able to diagnose fungal infections much better. And we may be able to say, okay, you have Candida in your urogenital tract, but don't worry about it. There's no need to treat this. Or you have it in there, and if we treat you, you're going to have less susceptibility to disease going forward. And that is just an amazing potential tool, just a diagnostic tool. And then on the treatment side, if we understand what those pathogenic strains are doing to cause disease using this in vitro model that we developed, wow, we could potentially use that information to block the host symptoms of the infection.
I love the aha moment you had. I think that's one you read about in books. I'm going to switch gears a little bit, and I want to talk about the University of Maine. Your background is like what mine was, which was, private universities for all sorts of different reasons, I'm sure, just like mine. And I've had the good fortune of tailing along with one of you guys in the academic world. So, I feel like I've been able to parallel watch what academics does for my whole life without actually having to do it. So it's kind of the ideal situation for me. And I didn't really appreciate how vastly important public universities are until my husband had a postdoc at UW. And then, ever since then, actually, he's been at a public university and our two kids, one of them went to the University of Michigan, right? I mean, he's been public school all the way. Anyway, was it a difficult culture change for you, or one that you were looking for to go from a background of private institutions into a public land grant institution where not necessarily a place where people look for basic research that you're doing at the molecular level because there's such a large focus on more immediate turnaround?
Wow. Well, there's lots and lots of things that I could say from there. I would say the first thing that was a big change for me was teaching undergraduates. And that's something that a lot of people have a big change from when they're a postdoctoral fellow. They're working really hard on the science, but probably not doing a lot of teaching, and then have to transition to a teaching role. Maybe half of the work that they do is teaching. And so that was a big transition. And I think that's a transition for most people who go even to a private university in a teaching role. So that was a big transition. And I was so happy to be at the University of Maine doing that transition. not only did I have fantastic teachers and mentors in the teaching aspects of what I was doing, but also the students are just so much more down to earth than the students that my previous professors and mentors had been working with in private colleges and universities, or private universities rather. The students that we have in the molecular biomedical sciences department, maybe I'm a little bit subjective about this, but there are some objective things about it. They are the best students at the University of Maine. We consistently have students from our department, salutatorian, valedictorian, the top of the college, and nationally competitive in grants and so on at the undergraduate level. So there's a lot of things to be proud about them, but at the same time, they are so well grounded in reality that many of them are first generation, and they have really developed a love for learning, in part because they have a parent or another family member who loves learning as well and maybe didn't have a chance to go to college. Or maybe they had a teacher or a couple of teachers that were really inspirational in elementary, middle, or high school who really inspired them to go to college. And they are coming at this love of learning from such a different perspective from a student who maybe had more privilege and always knew that they were going to go to college, and just college is a stepping stone to get to a professional career where they're earning six figures or more. And it's just such a pleasure to work with those students and try to identify their passions and enrich their ability to take advantage of that motivation and skill set that they've developed. So that has been one of the most wonderful things about working at the University of Maine. So let's get to the more difficult things. So one of the more difficult things working at the University of Maine is that I'm currently in a department with maybe 8 full-time professors. I think we may even be at 7 tenure track faculty members with strong research programs. But, all of them have strong research programs, which was not the case when I started at the University of Maine 15 years ago. So that department has really transformed in the last 15 years into a real engine for research growth. But nobody else studies fungi. I'm fortunate to have a couple of other faculty in the department that are interested in immunity and that also study zebrafish. So that's been really fantastic. And the study of zebrafish at the University of Maine, both in my department and other departments, has been so tremendous. We probably have the most number of zebrafish-focused researchers in biomedical sciences, the biggest concentration of any place in the world, almost one of the top 10 probably. So that's been really wonderful. But nobody else studies fungi. Nobody else studies really innate immune recognition of fungi or bacteria, for that matter, or viruses. That's not really something that people study. So what I've been really fortunate to have, as I mentioned, is this tremendous international community of collaborators that study Candida albicans and other fungal pathogens, and to be able to collaborate with them. So I have just really mostly been able to finesse that in a way that has been really positive. The university itself has not been focused on biomedical research, but there have been some more recent movement in that direction. The vice president of research has established an Institute of Medicine and has invested some real money into promoting biomedical research and also medicine in general, nursing that goes all the way from nursing to social work to aging and engineering sides of medicine as well. And the Graduate School of Biomedical Sciences and Engineering has been a great pull there. That Graduate School of Biomedical Sciences and Engineering has been a great way to connect individuals around the state of Maine as well and provide scientific environment and collaboration. So that's been really a great way to finesse that as well. But yeah, we've done a lot of convincing of the University of Maine administration that we have the potential to be even much greater than we are. And also that understanding basic biomedical science can be of very practical importance. And we saw this really in the work that was done with the Science Advisory Board that was advising the University of Maine system during the COVID pandemic, which included several basic scientists, Christy Townsend, Melissa McGinnis, Caitlin Howell, and myself, really focused on basic science, but having the ability to translate that basic science know-how into reading the literature and understanding the technology and understanding what's happening with the pandemic and being able to communicate that information both to the University of Maine system and to the general public. So I think really made a strong impression on the University of Maine administration and the University of Maine system in that those investments in biomedical science research over the last 20 years or so have really set us up for that ability to have a science advisory board with technical expertise.
I really appreciate you talking about, well, all of that, but the students and the love of learning. I think it is really easy to forget that education and learning in and of itself is super valuable for us as a whole. And that, the more that people embrace that and find their passion, even if we can't slot it into an exact, you're going to work in this field kind of thing, I still think we're better off. I have found, certainly when I've talked to faculty at UMaine, but also students, just the wealth of opportunities and this deeply collaborative nature between faculty and students at every level. It's really cool. And it wasn't necessarily my experience in undergrad. Not to slam, I don't want to dismiss what either one of our experiences were with these other institutions. I loved where I went to school. It was really great for me. It's wildly reassuring to see a place where education is fostered at every level. I have yet to run into a place where undergraduates have the opportunities presented to them that they have at the University of Maine.
Yeah, I would say it's really, we've been able to do a remarkable job at both prioritizing undergraduate education and also having strong biomedical research programs that provide those students with really top-notch, nationally competitive research programs in which to participate in. It's really, I would say, a labor of love among the faculty in our department and related groups to provide those research opportunities to undergraduates, and at the same time provide strong teaching to those undergraduates, and then have a strong graduate research program and overall research program.
Well, I think you're laying a foundation, not just, in fungal research, which, as an aside, funguses scare me more than probably anything else out there. I just think there's so much we don't know. I'm like, that's where the apocalypse is coming. It's from fungus. I also think you're laying the groundwork for a really solid research foundation for Maine moving forward, right? I've talked to a bunch of people, and it's certainly genetics and biology and microbiology in particular feels very much like rocket science in the 50s to me, right? Like, there's just so much that we are learning about. I cannot wait to circle back in 10 years to everybody I've talked to and be like, could you believe it? Can you believe that we've hit this, right? And that's what it seems to me that you're doing.
Well, we certainly feel that way. And just looking back 10, 15 years, it's amazing where we've come. So we have a lot of things that we want to do going forward, and we're excited. Yeah.
Well, I'm really glad that you're doing it. I'm glad someone is looking into fungus so that perhaps my paranoia will be a little less moving forward. This has been just a joy to talk to you, Rob. I have so many more things. I'm going to figure out a way to talk to you, whether it's for a podcast or not, to just kind of get some of my questions answered, because this was a really great conversation. Thank you.
Yeah, it was my pleasure. I really enjoyed talking with you. Great questions.
Thanks.
Thanks for listening to the Maine Science Podcast. Make sure you never miss an episode by subscribing on your podcast platform of choice. And please leave a rating and review. It will help more people find us and help spread the word about some of the remarkable people doing science in Maine. The Maine Science Podcast is recorded at Discovery Studios at the Maine Discovery Museum in Bangor, Maine. The Maine Science Podcast is executive produced and hosted by me, Kate Dickerson, and edited and produced by Scott Loiselle. The Discover Maine theme was composed and performed by Nick Parker.